IB Bio Animal Physiology Topic 11, Part 4, Movement B, will have its focus on the anatomy of the muscle fiber, the muscle cell. Once we fully understand the anatomy of the muscle fiber, we can move on to Part 5, Movement C, that focuses on how fibers contract, muscle physiology. This movie introduces a lot of vocabulary. Be ready to grapple with all the vocabulary. I will present imagery that initially zooms in, then zooms out, then zooms in again. Pay close attention. The essential idea is the roles of the musculoskeletal system are movement, support, and protection. Here is the outline of all the available movies for Topic 11. Use this outline to find the movie you need for review. This movie is focused here. Here are the first two IB syllabus statements of the movie. Skeletal muscle fibers are multinucleate and contain specialized endoplasmic reticulum. You should remember from Part 3, Movement A, that a muscle fiber is synonymous with muscle cell. Muscle cells are multinucleate, and this is an exception to cell theory. Secondly, muscle fibers contain many myofibrils. Myofibrils are composed of protein filaments, known as actin and myosin. The movement of the body involves skeletal muscle. In this image of skeletal muscle, we're looking at one cell or part of one cell. Within the cell, we see many nuclei. The striations seen here represent an underlying pattern of overlapping protein filaments that becomes very important in understanding how muscle fibers contract, how they shorten in length. You should recall that you've been exposed to striated muscle earlier in this course. Striated muscle is an exception to cell theory because striated muscle cells are multinucleate. Here is a significant IB syllabus statement. Describe the structure of striated muscle fibers, including the myofibrils with light and dark bands, mitochondria, the sarcoplasmic reticulum, nuclei, and the sarcolemma. You can see the vocabulary bursting out. Be patient, pay close attention, take notes. Let's start by looking at whole muscle, bundles of muscle cells, and then one fiber, one cell. A cross-section of this looks like this under a microscope. Again, we can see whole muscle. Then we have a bundle of cells with blood vessels. This is one cell here, and within the cell are numerous myofibrils, which in themselves are bundles of protein filaments, actin and myosin. Here is an actual micrograph of a single muscle cell, a single muscle fiber in cross-section. Notice all the myofibrils. Muscle fibers, single cells, contain many myofibrils, and the myofibrils are composed of protein myofilaments, long, slender protein molecules known as actin and myosin. Let's zoom back out just a bit and look at a muscle fiber, single cell. The cell membrane surrounds the cell, but the cell membrane of a muscle fiber is called a sarcolemma. Apologies for all the vocabulary. You can see that the cell is multinucleate. You can see the striations. The alternating light-dark bands represent locations within the cell where the actin and myosin filaments overlap, the dark, or do not overlap, the light. More on this as we move along. Once again, here is a micrograph of a muscle fiber. We can imagine the cell membrane, the sarcolemma. Notice the multinucleate nature of the cell. Also within the cell is a specialized endoplasmic reticulum called sarcoplasmic reticulum. In this diagram we have a whole muscle, and this is one cell, one fiber. The sarcolemma is the cell membrane. The sarcoplasma is the cytoplasm. You can see the many nuclei, and these lines represent myofibrils. Now remember, we're not highly magnified here. Each myofibril is actually composed of numerous protein filaments, actin and myosin. You've seen this image before. A whole muscle is cut in cross-section to illuminate muscle structure. A single cell is extended to show multiple nuclei and many myofibrils. The membranes that surround each myofibril is the sarcoplasmic reticulum. It's the specialized endoplasmic reticulum, and you need to look carefully to see it. You can see how each myofibril is composed of myofilaments, proteins, actin, and myosin. And if we look closely right here, we can see how the actin and myosin overlap and then do not overlap. The alternating overlap and non-overlap of actin and myosin produces the striations, seen here. I'll be showing you more highly magnified images of all these structures. Pay close attention and be patient. Here is a close-up image of a muscle fiber. Within the sarcolemma, the cell membrane, are numerous myofibrils. And these are composed of filaments, actin and myosin, that can only be seen in cross-section here. You can see a nucleus, it would be one of many, and numerous mitochondria can be seen, and this should make sense because muscles spend a lot of ATP to contract. The specialized endoplasmic reticulum, known as sarcoplasmic reticulum, is seen here, and T-tubules connect the surface membrane, the sarcolemma, with the sarcoplasmic reticulum. And here you can get the idea of actin and myosin overlapping or not, overlapping, not. So this image is even more close up on a muscle fiber. Here's the cell membrane, the sarcolemma. Here's the sarcoplasmic reticulum, the specialized endoplasmic reticulum. And here is a myofibril. This image better illustrates the actin and myosin protein filaments, and it provides a new term, the sarcomere, that I'll clarify shortly. This is a myofibril, and we see the actin and myosin filaments lying parallel to one another, and they overlap in some regions, but not others. The overlapping filaments produces the striations that we see when we examine muscle cells under a microscope. The thick protein is the myosin, and the thin protein filaments is the actin. Now, the sarcomere is the functional unit of a muscle cell. The sarcomere is defined from Z line to Z line, and it is the structure that shortens or contracts. The myosin and the actin slide past one another to effect shortening. You can see that there are terms related to these overlapping filaments. The length of the myosin filament is known as the A band, and the region with actin only is called the I band. Be patient. Take it one slide at a time for understanding. Here is an image of a fiber. The sarcolemma is here. Numerous myofibrils can be seen, and muscle fibers have numerous mitochondria to provide the necessary ATP for contraction. Now, if we isolate a single myofibril composed of actin, blue filaments, and myosin, red filaments, you can see regions of overlap and regions where the filaments do not overlap. These alternating regions produce the striations that we see when we look at muscle with a light microscope. Form, function. The form, the pattern ties directly to functions. The striations result from the overlapping actin and myosin filaments within the cell. The length of the myosin filament is known as the A band, and this is an area of high overlap. The actin-only region is known as the I band, no overlap. Here is a myofibril composed of actin and myosin filaments. You can see the alternating pattern of the filaments, no overlap, overlap, no overlap, etc. A sarcomere is defined as the contractile unit of a muscle cell, Z line to Z line. Be patient, I'll work to clarify all terms. More highly magnified, here is the sarcomere, defined from Z line to Z line. The sarcomere has actin, myosin, filaments. The myosin is the thick filament with heads, known as cross bridges, that have the enzymatic role of breaking ATP. Actin is the thin filament, and actin is anchored to the Z line. Scientists have defined the bands. The I band is a region of actin only. The A band is the length of the myosin filament. The H zone, not annotated here, is the myosin-only region. The contraction of skeletal muscle is achieved by the sliding of actin filaments toward the middle of the sarcomere, the sliding of actin across the myosin. Here is a nice image providing a three-dimensional look at the sarcomere. You can see the thin actin filaments anchored to the Z line, a protein structure. You can see the thick myosin filaments in the center of the sarcomere. What's nice about this image is that you can now imagine how a sarcomere shortens, how it contracts. The actin filaments slide to the middle of the sarcomere. The myosin heads, seen in here, bind the actin and pull the actin inwards. The contraction of skeletal muscle is achieved by the sliding of actin and myosin filaments across each other. The sarcomere shortens when the myosin cross bridges bind to the actin and pull the actin toward the middle of the sarcomere. The action of contraction requires ATP. Stay tuned. At this point, you should be able to order muscle structures from largest to smallest. We have the whole muscle, the fiber, the myofibril, the sarcomere, the contractile unit of muscle, myofilaments, myosin and actin, and then structures to which you have not yet been introduced, tropomyosin and troponin. These proteins regulate contraction. The physiology of contraction will be covered in Part 5, Movement C. Do you have control of these IB syllabus statements? Describe the structure of the contractile sarcomere. Draw a labeled diagram of the structure of a sarcomere, including Z lines, actin filaments, myosin filaments with heads, and the resultant light and dark bands. These are the striations that I've referred to. Here is a relatively unmagnified micrograph of sarcomeres. We can see a sarcomere here, Z line to Z line. The I band is here, actin only. The A band is here, it's the length of the myosin filament. The H zone, barely visible, is myosin only here, and again this is I band here. Here's a nice image to study because it displays a micrograph of a sarcomere at the top and the corresponding diagram below. So here is our sarcomere, Z line to Z line, Z line to Z line. The A band is the length of the thick myosin filament. The I bands, here and here, are the actin-only portions of a sarcomere when the sarcomere is not contracted. Keep that in mind. The H zone at the center of the sarcomere is the myosin-only portion of the sarcomere when the sarcomere is not contracted. The contraction of skeletal muscle is achieved by the sliding of actin to the middle of the sarcomere past the myosin filaments. The myosin cross bridges actively pull the actin toward the center of the sarcomere, increasing the overlap of actin and myosin. Again, here is a micrograph of a sarcomere, Z line to Z line, and here is the corresponding diagram that defines what it is we're looking at, Z line to Z line. The I band is a light region, actin only. The A band is a dark region defined as the length of the myosin filament, and the H zone is myosin only. As we look at sarcomeres in longitudinal sections, as we've been doing, we must remember that they are three-dimensional structures. In this diagram, we can see the sarcomere, Z line to Z line. The Z lines are protein that anchors the actin. We can see the thin actin filaments, and we can see the thick myosin filaments. Here is the I band on each side of the sarcomere, and here is the A band, and here is the H zone. In thinking about the three-dimensional quality of the sarcomere, imagine cross-sections through three different portions of the sarcomere. If we were to examine a cross-section through the I band, we would see actin only. A cross-section through the H zone would result in myosin only, and a cross-section here would result in a combination of actin and myosin filaments. So here are relevant IB syllabus statements. You may already have good control of these two statements. The contraction of skeletal muscle is achieved by the sliding of actin and myosin filaments. Analyze electron micrographs to find the state of contraction of muscle fibers. So here is your sarcomere, Z line to Z line. You can see the myosin and the myosin heads, the cross bridges that project toward the actin. As contraction happens, the actin is pulled to the middle of the sarcomere by the myosin heads. Let's take a look. On this slide, the top diagram is an uncontracted sarcomere, Z line to Z line, while the bottom diagram displays a contracted sarcomere. Notice the distance between the Z lines in the contracted sarcomere is significantly shorter than the distance between the Z lines in the uncontracted sarcomere. Now, the uncontracted sarcomere has an I band here and here. A band is here, and here is the H zone. As the sarcomere contracts, the actin is pulled to the center of the sarcomere by the myosin heads. The overlap between actin and myosin increases in the contracted sarcomere relative to the uncontracted sarcomere. The I band decreases, as does the H zone. The A band, which is defined as the length of the myosin, does not change length. In its contracted state, seen here, the sarcomere approximates the length of the A band. The I bands and the H zone are nearly absent. Once again, at the top is a sarcomere in an uncontracted state. Notice the width of the I bands and the H zone. Notice the length of the A band. The A band is the length of the myosin filament, and notice the myosin heads protruding from the myosin. In the contracted state below, the actin has been pulled to the middle of the sarcomere by the myosin heads. The myosin heads, the cross bridges, require ATP. Once contracted, the sarcomere I bands and H zone have nearly disappeared. The contraction of skeletal muscle is achieved by the sliding of actin and myosin filaments. One could measure the change in the length of the sarcomere upon contraction. And here's the relevant IB syllabus statement. Measurement of the length of sarcomeres will require calibration of the eyepiece scale of the microscope. In this micrograph of a sarcomere, we have a scale line provided. You can see the width of the full I band, each side of the Z line, is about 0.5 micrometers. I've used this scale line to define the length of the sarcomere as approximately 1.75 micrometers. In this micrograph, the top image is an uncontracted sarcomere, Z line to Z line, and the scale for the I band on either side of this Z line is given as 0.5 micrometers. In the lower micrograph, the sarcomere is contracted, and the I band on either side of the Z line has been reduced to 0.25 micrometers. In this micrograph of two muscle sarcomeres, one here and one here, we can see the change in the length of the sarcomeres from the uncontracted to the contracted state. Just take note of the distance between the Z lines in the uncontracted state and the Z lines in the contracted state if you're not sure that sarcomeres truly shorten. In the uncontracted state, we can see distinct I bands and an H zone. I bands and an H zone. Once contracted through here, there really is no H zone, and the I bands are far less distinct, as the actin has been pulled to the middle of the sarcomere. Once contracted, each sarcomere is the approximate length of the A band, the length of the myosin filament. Here is a chance to practice. Is this sarcomere contracted or uncontracted? Turn the movie off now, or allow me to say that it is uncontracted. I bands and the H zone are clearly apparent. This micrograph is nice to study because the thick myosin filaments are distinct from the thin actin filaments. The I and A bands are labeled along with the H zone, and if you look really closely, you can almost make out the myosin heads, the cross bridges that lie along the myosin filament, bind to the actin, and draw the actin filaments to the center of the sarcomere. Here is a very highly magnified micrograph of a sarcomere. It's so highly magnified that we can only see a partial sarcomere. I've labeled the actin and the myosin. The H zone is here. You can see the myosin heads through this region, a region that lies between the myosin and the actin. Remember, the myosin heads pull the actin to the center of the sarcomere, causing shortening. If you feel you already understand how muscles function to achieve contraction, you're right. But there are some further biochemical details required from the IB syllabus. These details are the subject of the next movie, Part 5, Movement C. Here's a chance to practice your knowledge of sarcomere structure. You can turn the movie off now or follow to the next slide. You can study this slide on your own, but you should ask yourself, is this sarcomere contracted or relaxed? It's relaxed. Do you know why? As we get ready to bring this movie to a close, here is the first of 3 micrographs to help you understand the anatomy of a muscle cell that includes the sarcomere, here, the sarcoplasmic reticulum, and T-tubules. Go back to the beginning of this movie if you've completely forgotten the sarcoplasmic reticulum and T-tubules. In this image, we can see the sarcomere, Z-line to Z-line. The full I band on either side of the Z-line is here, and the A band is here, and we can see the H zone. It appears that this sarcomere is not contracted. Numerous mitochondria, labeled M, can be seen, and the arrows point to T-tubules and sarcoplasmic reticulum. T-tubules are inward extensions of the cell membrane, the sarcolemma, that extend deep into the cell from the cell surface. Here is a micrograph showing the sarcomere, Z-lines are here, with mitochondrion and T-tubules and sarcoplasmic reticulum. Here is an excellent image to study because it is quite well labeled. Here is the sarcomere, Z-line to Z-line. Here's the I band. Here's the A band and the H zone. Here's the sarcoplasmic reticulum and the T-tubule. We can see glycogen here and here and here. And this image nicely combines the micrograph with a diagram of what we're looking at here. Spend some time studying this micrograph. Nerve cells send signals to muscles to cause contraction. The depolarization of the sarcolemma, the muscle cell membrane, causes depolarization of the membrane of the sarcoplasmic reticulum. Keep this in mind, it's the T-tubules that serve here. Remember that muscle cells have special endoplasmic reticulum called sarcoplasmic reticulum. The sarcoplasmic reticulum, once depolarized, releases calcium ions into the cell cytoplasm. And it's the presence of calcium ions that initiates sarcomere contraction. On to Part 5, Movement C. And that brings us to the end of IB Bio Animal Physiology, Topic 11, Part 4, Movement B.